So in this experiment, we shall observe stomata of this leaf with help of this microscope. So these are the materials which we need. Saffron in solution for staining, distilled water, watch glass, and 2 clean slides and cover slips. We shall prepare the slide. We will prepare a temporary mount of leaf epidermis over this slide. First, some water in watch glass, and then we know that stomata are present on the dorsal side, opposite side of the surface. So to cut out the epidermis, we will cut the leaf like this, and we can see the epidermis of dorsal side comes out. You can see. Alright, we shall cut this epidermis. And we shall put this epidermis in water with help of brush. जो एपिडर्मिस है वो अभी वाटर में है. Now we shall take saffron in solution and put a few drops in this watch glass carefully to stain the epidermis. We shall mix this saffron in solution to stain this epidermis. Wait for 1 or 2 minutes. So we can see that epidermis is now stained with saffron in solution. Now we shall take a clean slide and with the help of brush place this epidermis over the slide carefully. Remove any air bubbles if it has. So you can see the slide. Now we shall remove the excess water with help of filter paper. Filter paper will absorb the water like this. Now we shall put a cover slip. Cover slip is like this, a small cover slip, carefully with the help of needle, so that air bubbles do not form. We will put like this with the help of needle and slowly put the slide over the epidermis. So our slide is ready. We shall remove excess water with help of filter paper, and we can see there are no air bubbles in this slide. We can also add some glycerin for clarity, but it's optional. So now we shall observe this slide on this microscope. To know the working of microscope and its parts, you can watch previous videos, previous classes. So as I had told you before, this is the eyepiece from which you see, and these are the objectives, 2 lenses of different magnifications. This is the mirror for light reflection, and these are the focuses. So first of all, we have to reflect the light here on this lens so that we can see clearly. So we shall first adjust this mirror so that we can see clear light, white light. Okay. First we shall use magnification, small magnification of eyepiece and objective. 10 multiply, there are 2 lenses, 10 multiply and 15 multiply. First we shall use lower magnification, then we shall observe with higher magnification. So at this moment there is no slide, but we should have clear light over this lens. You can see here is clear white light. You can see it is very light. This is very bright light. Okay. Now we shall put this slide over the bench of this microscope and observe from eyepiece until we can see a clear image. So this is the main focus, this is fine focus. First we shall move the main focus. Yes, now I can see clearly, and then I will move fine focus for more clarity. Right. Okay. So I can see stomata. I will show you. So I will try to show you what can be seen through this eyepiece. You will see very small sized of stomata. If you can see now, what you are seeing at this moment, this is plant cell, and there are stomata in between, guard cells, stomatas surrounded by guard cells. Okay. Now we will see through larger magnification. Okay, this is 10 multiply, we shall use 15 times magnification and adjust the focus. Yes. So under higher magnification, let's see. You can see bigger size guard cells surrounded by chloroplast with stomata. Stomata surrounded by guard cells. Here in white color, we can see stomata with guard cells. Now we shall move this eyepiece and observe it under higher magnification. So if you can see a guard cell with stomata here in middle, if you can see clearly, okay, this is one stomata with guard cells. Let's see again. As you can see, this is also stomata with guard cells. So that's how we see stomata under microscope.